Hello everyone, my name is Peter Nguyen and I'm an undergraduate student at the University of California, Santa Cruz. Today, I am going to talk about the effects of drought and warming on Lupinus nipomensis' survival and reproduction through ecophysiology and eDNA techniques. Lupinus nipomensis is an endangered plant native to the Guadalupe-Nipomo Dunes in San Luis Obispo. It has a variable interannual population. Because of this, we are interested in how drought and warming influence its growth and reproduction. We are also interested if there are microbial communities with particular microhabitats or climate manipulations. In the first portion of our study, we grew 80 individuals under 4 treatments: drought, warming, and control over a 6-month period. We grew these individuals in a greenhouse to isolate for the variables we're studying for. Warming was applied using a one-sided open-face polycarbonate cube and heat lamp, which raised the temperature about 4 degrees Celsius. Episodic drought was implemented and the plants were re-watered when stomatal conductance was 0.05 moles per meter squared per second. We recorded morphological, physiological, and leaf traits. For the second portion of our study, we collected soil samples from 4 treatments in our original greenhouse study and samples from 4 treatments from a field study site in San Luis Obispo. We identified 4 plots from the field site to take eDNA samples from: wet-cool with reproductive success and no success, dry-warm with success and no success. The main takeaways from our statistical analysis of the ecophysiology biometrics were: flowers were further from the base in drought and warming treatments, warming increased while drought decreased stem and leaves, drought decreased seed pods but increased reproductive efficiency. Furthermore, drought had a significant effect on photosynthetic rates and stomatal conductance through all time periods but the last drought period. Droughted individuals had a higher water use efficiency compared to well-watered plants. Things to consider from the study are that Lupinus nipomensis showed an increase in water use efficiency and reproduction in response to drought, indicating drought adaptation. Warming produced more leaves and stems, which increased plant water requirements, which could increase drought stress in dry years. Warming increased most plant traits, which could be related to their earlier flowering and death times. Finally, droughts lowered seed pod production and greater reproductive efficiency in self-pollination is concerning because successive drought years can lead to low seed banks. For the eDNA portion of our study, we are currently extracting DNA from these samples and are performing meta-barcoding to identify possible important microbial communities. If successful microbial communities are found, soil transplants or plant relocation can be done to help in the species recovery. Furthermore, if this conservation model is successful, we can innovate biodiversity conservation by applying this restoration model to other species. With that, I'd like to thank you for coming to my lightning talk, and I'm happy to take questions now or later through email.